Hello, everyone, and welcome back. Thank you for joining us. I'm Steve,
And I'm Scott.
and together we're Backyard Musings. I'm broadcasting from the People's Republic of California in Apple Valley.
And I'm up in Utah, in the bunker.
There you go, on assignment.
And we're talking science technology, and we got a great story for you today.
Yeah, this is a good feel-good story. A study that compared the gene expression profile of Alzheimer's disease with those triggered by 1,300 approved drugs identified. Again, this is AI. Identified a combination of two cancer medications as a potential treatment for the most common form of dementia. Researchers from UC San Francisco and the Gladstone Institutes have discovered that certain cancer medications may help counteract the brain changes caused by Alzheimer's disease. This breakthrough could lead to treatments that slow or potentially reverse the condition's symptoms. The findings appeared in the journal Cell on July 21st. Would you like to know more?
Yeah, again, this is one of those deals where sign me up, right? If I'm showing signs, or I got a parent that's showing signs, or whoever, or a spouse, I'm sign me up. What do you got to lose, right?
Right.
To begin, the scientists examined how Alzheimer's alters gene activity in individual brain cells. They then searched for FDA-approved drugs that produce the opposite effect on gene expression. Their focus was on medications that could correct changes in neurons and glial
Leo.
cells, both of which are disrupted during the progression of Alzheimer's. The team also reviewed millions of anonymized medical records and found that people who had taken some of these drugs for unrelated health issues were less likely to develop Alzheimer's.
Finally, when the researchers tested the two most promising drugs, both originally developed to treat cancer in mice with Alzheimer's like symptoms, I don't know how mice get Alzheimer's, I have no idea. The results were striking. The treatment reduced brain damage and helped restore memory function in the animals.
Amazing.
Good for the mice.
Quote, yeah, quote, Alzheimer's disease comes with complex changes to the brain, which has made it tough to study and treat. But our computational tools opened up the possibility of tackling the complexity directly, says Marina Sirota, PhD, the interim director of the UCSF. I'm going to assume that's probably University of California, San Francisco, right? Bakar Computational Health Sciences Institute, professor of pediatrics and co-science author of the paper. Go on with the quote. We're excited that our computational approach led us to a potential combination therapy for Alzheimer's based on existing FDA-approved medications. So that's good.
Yeah, so sometimes medications have a dual purpose, like
Ivermectin.
ivermectin. Yeah, that's a good example. I was going to say, bupropion has a function for depression, but it's also a stimulant that helps with focus. It's a two-part thing that helps.
I need to write that down.
Yeah. The research was funded in part by the National Institutes of Health and the National Science Foundation. Alzheimer's disease affects 7 million people in the U.S. and causes a relentless decline in cognition, learning, and memory. Yet decades of research have only produced two FDA-approved drugs, neither of which can meaningfully slow this decline.
Okay, another quote. Alzheimer's disease comes... Oh, no, I already read that one. Alzheimer's is likely the result of numerous alterations in many genes and proteins that together disrupt brain health, said Yadong Huang, MD, PhD, senior investigator, and director of the Center for Translational Advancement at Gladstone. Also professor of neurology and pathology at, again, UCSF, and co-senior author of the paper. Another quote, this makes it very challenging for drug development, which traditionally produces one drug for a single gene or protein that drives disease. So
the team took publicity. Go ahead, Scott.
No, I was going to say that. I guess that's right. They're all trying to pinpoint, like we're just saying, they're all trying to pinpoint the one issue, but maybe there's something out there that that's already in practice that they could tweak a little bit or whatever, and or combine it with another drug.
I'm telling you, this was AI. They taught the AI what to look for, and then it went out and looked at all the FDA approved drugs. I'm telling you, AI is powerful.
Yeah.
The team took publicly available data from 3 studies of the Alzheimer's brain that measured single cell gene expression in brain cells from deceased donors with or without Alzheimer's disease. They used this data to produce gene expression signatures for Alzheimer's disease in neurons and glia. The researchers compared these signatures with those found in the connectivity map, a database of results from testing the effects of thousands of drugs on gene expression in human cells.
Out of 1,300 drugs, 86 reversed the Alzheimer's disease gene expression signature in one cell type. 25 reversed the signature in several cell types in the brain, but just 10 had already been approved by the FDA for use in humans. Pouring through records housed in the UC Health Data Warehouse, which includes anonymized health information on 1.4 million people over the age of 65, the group found that several of these drugs seem to have reduced the risk of developing Alzheimer's disease over time. Sounds like a pretty good stat.
Yeah. Here's a quote. Thanks to all these existing data sources, we went from 1,300 drugs to 86 to 10 to just 5, said Yakio Lee, PhD, a former UCSF graduate student in Sirota's lab, who is now a postdoctoral scholar in Huang's lab at the Gladstone and the lead author of the paper. Quote, in particular, the rich data collected by all the UC health centers pointed us straight to the most promising drugs. It's kind of like a mock clinical trial.
Yeah. Li, Huang, and Sirota chose 2 cancer drugs out of the top 5 drug candidates for laboratory testing. They predicted one drug, letrozole, would remedy Alzheimer's in neurons, and another, irinotecan,
Yep.
Okay, would help glia. Letrozole is usually used to treat breast cancer. Irinotecan is usually used to treat colon and lung cancer. Okay.
The team used a mouse model of aggressive Alzheimer's disease with multiple disease-related mutations. As the mice aged, symptoms resembling Alzheimer's emerged, and they were treated with one or both drugs.
The combination of the 2 cancer drugs reversed multiple aspects of Alzheimer's in the animal model. It undid the gene expression signatures in neurons and glia that had emerged as the disease progressed. It reduced both the formation of toxic clumps of proteins and brain degeneration, and importantly, it restored memory.
Yeah, so, that's awesome.
Sounds like they're onto something here.
So, right now, you can go to your doctor for your loved one and say, hey, let's try these 2 drugs and see what it does for grandma or grandpa or whoever is experiencing this. And see if it works. It's not like it's got to go through clinical trials. It's a drug already out there. Just get it and use it.
And I'm going to just throw a question out there. Why are doctors, now that these studies have been made, why are there doctors out there that are not treating people with dementia or Alzheimer's, saying, hey, let's give this a shot and see what happens?
Well, this is cutting edge. This is probably it was just published in Cell, and so now that the doctors got it, now we should see this happening and see the results. Doctors should start talking about it.
Yeah, absolutely. So yeah, hopefully they're sharing this stuff. So
really good story.
All right. Yeah, it is a good story. I mean, I've known several people that had this, and a lot of them have passed on, and I know some people that are going through it right now, and this would be game changing. I think be cool.
Yeah.
So
all right, folks, thanks for joining us. See you this afternoon.
Take care, everyone. Thanks.